Hello, BISC 210 students. Today we're going to be covering Chapter 7, which includes microbial growth, controlling microbial growth. We'll also talk about media, how we grow bacteria in the lab, for instance, which is pretty relevant to what you're going to be seeing when you get your laboratory kits. So let's get started. All right. So when conditions are favorable, microbial growth occurs, depending upon the organism, pretty rapidly. And the number of total cells in that population will increase quite a bit. But that's under favorable conditions. Now, conditions aren't always favorable. So with microbial growth, in favorable conditions, you have more microbes growing at a faster rate. A really good example of this is E. coli. In the lab, under really favorable conditions, we can get E. coli to grow, replicate every 20 minutes or so, 20 to 24 minutes. We get it to go through a round of binary fission, and the population doubles every 20 to 24 minutes. Whereas other organisms like tuberculosis, Mycobacterium tuberculosis, has what we call a generation time of 16 to 18 hours. So it doesn't undergo binary fission for about 16 to 18 hours. So you can imagine the total number of cells in that population slowly goes up over time compared to that of a faster growing organism like E. coli. So nature versus nurture, or nature versus the lab, natural versus the lab. In the lab, we get pure cultures, meaning when we plate samples, we use different types of media. We can get pure cultures, meaning isolated colonies. And this is something you guys saw in the videos in lab from your TAs, streaking for isolation. And the point was to dilute the sample so eventually you get pure cultures. However, in nature, we really don't get pure cultures. If you were to swab pretty much any surface that hasn't been cleaned or disinfected or sterilized, you're probably going to get a mixture of a bunch of things. And you saw that with some of the plates that I posted in Lab 4 folder. There's a plate that had both fungi and bacteria on it, and they came from the bottom of your shoe, or a shoe. So it goes to show you that everything is kind of intermingled. Environmental factors play a really important role in the life and metabolism of bacteria. Some organisms pick up new traits. Some are different. They're what we call different serotypes or different versions. So, for instance, when I say E. coli, granted, that is the name of a bacteria, but there are so many subgroups that you can have an E. coli that doesn't have one version that doesn't have flagella. You can have an E. coli that one version has flagella. You can have E. coli that doesn't cause disease. You can have E. coli that does cause disease. So there are different versions. When we get to diseases of digestive system as well as the urinary system, E. coli 0157H7, those numbers I know right now don't mean anything to you, but that is the most pathogenic strain, disease-causing strain of E. coli. But you can see the numbers and letters after the name indicate what version it is. So a lot of people are under the assumption that if I say, okay, well, we're going to work with E. coli, there's only one type of E. coli. Or they have an infection with Streptococcus pyogenes, which causes strep throat. It's only one version. That's not the case. There are multiple versions depending upon the organism. And it can be quite significant, like E. coli. Dynamic and complex growth in nature One of those would be biofilms. Biofilms are really cool. Basically, what they do is the glycocalyx allows the microbes to be sticky, and they will attach to a surface, and they're really hard to get rid of. Okay, they're really, really hard to get rid of. You experience a biofilm every single day. You may not think about it that way, but you do. And I'll get to what that is in a second. Biofilms are really hard to treat in healthcare setting because the bacteria are stacked on top of each other. The sugars make them really sticky. It's hard to actually get them off of a surface. And that's why persistent infections are pretty common with biofilms. A great example of that is a patient who uses a catheter, what we call an indwelling catheter. Basically, an indwelling catheter is a catheter that is in place for extended periods of time, okay, longer than a day, several days, a week. What can happen is biofilms can form, and then they travel up the urethra and into the bladder, causing infections in the bladder, and then all the way up to the kidneys eventually, which causes a whole host of problems. Biofilms are really hard to treat. The biofilms you form, or your body that form, is brush your teeth in the morning. Okay, make sure nice and clean, and then before you eat lunch, before you eat anything actually, take your fingernail and scrape the front two teeth on your in your mouth. And that film, that white film that forms on your fingernail, is a biofilm. Now, granted, there's going to be some food particles in there and some other stuff. Most of that is bacteria. Okay, there's tons of bacteria in your mouth, and basically tons of bacteria in your mouth, and they will form these biofilms. We talked about binary fission already. Basically, binary fission happens in prokaryotic organisms. You copy the DNA, each cell gets an exact copy, and then it's an asexual process. Generation time. So this is something I mentioned before. Generation time, this is the amount of time it takes for a cell to go through binary fission. And times are really diverse. You can see ranging from 15 minutes to 24 hours. It really depends upon the species and conditions. And there are a lot of different growth factors that affect the rate of the reaction. So, depending upon what you're looking at, will determine whether or not, and what's in the environment will determine whether or not the generation time is faster or slower. If you were to take E. coli, put it in media in the lab, provide it with an optimal temperature and optimal pH, it will replicate very, very quickly. But if you cool it down, if you don't give it as much chemical growth factors or physical growth factors, then it's going to take longer to go through binary fission. So there's lots of things that affect time, generation time, and growth. We're going to talk about some of these examples. We can calculate generation time, which is really neat on how to do that. So in order to do that, we have to understand that bacteria grow in a very similar way. Now, what I mean by that is there is a growth curve that most bacteria follow. And that growth curve I'll show you in the next slide. But I want to show you this first before we talk about that growth curve. Now, there's a phase in the growth curve called exponential growth. And this is where most of the bacterial growth is going to happen. So the question here is, E. coli are grown for 1 hour. During that time, they go through 3 generations of growth. What is the generation time? So to determine generation time, which you have to calculate in minutes, you take the growth time in minutes divided by the number of generations. So in this case, 1 hour is 60 minutes, and there were 3 generations. So we divide 60 minutes by 3, and our generation time is 20 minutes. So what does this mean? It means that every 20 minutes, the population doubles. And you can see here, so if we started with 1, 20 minutes went by, now we have 2. Another 20 minutes went by, we have 4. And then from there, we have the population doubling again to 8. So, and then from there, so on and so forth. So you can see that generation time tells us how long it takes for that organism to replicate and increase the number of cells in that population. Make sure you know this formula. I guarantee you'll see questions about it at some point. This is what I was talking about in terms of growth curves. There are 4 major stages of the growth phase, or the growth curve, growth phase, whatever you want to call it. Either term is acceptable. You have the lag phase, which is when they're starting to establish their, the bacteria is starting to establish itself in that environment. Then, if there's favorable conditions, you get exponential growth, what we call logarithmic growth, where most of the growth is happening. And this is where you're calculating your generation time, because the population is going to double every however minutes the generation time is. So lots of them go up, 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 up really fast until they plateau at the stationary phase. Now, my question to you is, why do we get a stationary phase? Why at some point does the cells in this, whatever, let's say, vial of media hit this stationary phase? Why doesn't logarithmic growth just keep going and not go nonstop? What you have to take into account is, let's say we're growing it in a lab, and we're growing it in 100 milliliters of broth, nutrient broth. And logarithmic growth occurs, eventually it hits stationary phase. Why does it hit stationary phase? Well, at some point, as the population grows, they're going to run out of resources, right? Space is going to become limited. The access to growth factors like nutrients will become limited. What else? Oxygen availability. And then most bacteria, once they go through metabolic functions, they produce a waste product. And in that vial, that waste product, all those cells are producing some form of a waste product. And basically, once that waste product gets to be too high, there's no way for the cells to continue to grow. Right? The population pretty much levels off because there's just it's not a favorable environment anymore. They're out of growth factors. They're out of, for instance, nutrients, space, oxygen, all those things. They're just out of there's just no more. So they hit the stationary phase. And then eventually, because all of those things are lacking, you then move into this 4th phase, the death phase. We also call this the logarithmic decline phase as well. Either way, death phase is totally acceptable. Knowing these 4 phases or stages is pretty important. Lag, log, stationary, and death. Now, another question is do you think this curve is the exact same for every organism? Do you think every curve follows it exactly this way for every organism? The answer is no. You would still have the 4 phases, but if you have something that has a long generation time, this would take longer, right? This would take much longer to get a large number of cells the generation time is 18 hours, its population does not double very quickly. So the actual graph will stretch or condense based on the growth of the organism, whether or not the organism is growing very quickly or pretty slowly. Now, on top of being able to calculate generation time, we can actually estimate a number of cells in a population. So we can actually estimate how many cells are in a given population. So what we do is we use a very specific formula. Total population equals 2 to the nth times the number of starting cells. N stands for the number of generations. N stands for the number of generations. It's the same thing if we go back to this right here, okay, growth time in minutes. We can use the letter N for generations. Okay, so you would put growth time over N, and N stands for the number of generations. So let's ask ourselves a question. Let's test this. We start out with 20 cells. How many are present after 8 generations? Well, with this information, we can determine whether or how many cells are approximately in this population. So 2 to the 8th, because we start out with 2 and then it goes to the 8th power. N is the number of generations. And then we multiply it by the starting number of cells. So 2 to the 8th is 256 times 20 gives us 5,120 cells. So roughly, if we start with 20 cells, how many are present after 8 generations? Approximately 5,100 cells. So we can use this information to calculate total population. Make sure you understand both the formula for generation time as well as the formula for total population. I guarantee you will see these at some point later on. So one of the growth factors is temperature. The lower, depending upon the organism, let's say we'll talk about organisms that typically grow in our body. That's around 37 Celsius, which is 98.6 roughly, 98-ish roughly. So 37 Celsius. That's really the optimal temperature for most organisms, and that's when they're going to thrive and do really well. If you were to drop that temperature, the bacteria would slow down. Their growth time would slow down. But if you stray too far above that temperature, then the proteins become denatured. We talked about proteins when we talked a little bit about macromolecules and how they're held together by hydrogen bonds. If hydrogen bonds are holding the protein structures together and we get too high of a temperature, the hydrogen bonds break and the protein denatures. Basically, it unwinds. Once that happens, there's no fixing it. That protein is destroyed. It no longer works. So we have to be very careful in terms of denaturation of proteins. There's a whole bunch of different organisms and within the ranges of temperature. So if we go back, you can see extreme thermophiles, mesophiles, psychotrophs, psychrophiles, all those fun things. But I only expect you to know 2. So, I only expect you to know these 2, psychotrophs and mesophiles, because of the range they live in temperature-wise, and what they're associated with. So psychotrophs, I don't expect you to memorize that they're 0 to 30 degrees Celsius. That's not really a big deal. But they are associated with foodborne illness. These are the organisms that you find typically in your fridge. So you leave food in your fridge for a while, and stuff starts growing. Psychotrophs, most of the time. Not all the time, most of the time. Psychro just means cold. This means cold. Mesophiles, this is around the middle ground of temperature, and this is where most pathogens, human pathogens, survive in this range. Because our body temperature is around 37 Celsius. So it's pretty much a little bit above the halfway point in this range. So mesophiles tend to be associated with most human pathogens. Another growth factor is oxygen requirements. Lots of microbes can live without oxygen or with minimal oxygen. Especially organisms that live in the soil. Good example of that is Clostridium. Clostridium tends to live in the soil and really has little access to oxygen. But they can still thrive perfectly. Most pathogens do live in low oxygen environments in the host, because if you think about it, let's say you get a pathogen, Shigella or Salmonella, that infects your small intestine, right? Your small intestine does not have a ton of oxygen, very little. So in your GI system, with very little oxygen present, most pathogens need to be able to adapt to that and survive. And they can. There are very few places within the body where there's a ton of oxygen, right? The lungs, sure. In your mouth, nose, there's lots of oxygen. But if you start talking about within a wound, the deeper the wound, the less oxygen that gets into it. The deeper the tissue, wound in the tissue, the deeper, the further you get into the gastrointestinal tract. Either way, you're going to start to see less and less oxygen. Atmospheric oxygen, O2, needs to be modified in order to be usable. So atmospheric oxygen, O2, can get across plasma membranes in terms of bacteria, but they need to be modified. So bacteria have to have two enzymes. Superoxide dismutase, I'll refer to it as SOD, which takes reactive superoxide ions and turns it to hydrogen peroxide. And then catalase will then convert the peroxide into water and oxygen. If you don't know the formula of peroxide, it's H2O2. So basically, you're taking H2O2 and breaking it into water and oxygen, which is pretty straightforward. Now, if an organism has both of these, you can expect them to survive in environments that are rich in oxygen. If they have one, if they only have one or the other, then maybe they can live in an environment with minimal oxygen. If they have neither, chances are they don't do well, they don't survive if they live in an environment with the presence of oxygen. So this table, table 7.1, this number is wrong. Table 7.1 is wrong in your book. Or yeah, in your book. This image is from the old textbook that I used to use, but I will find the number for you, the correct number, and post it on Blackboard when in the new textbook, the one that you guys are using. I just happen to like this image a lot, so I didn't switch it out. It's covered in your book as well, so it's not like it's something that's not there. It's just I prefer this image. So this is a really great image talking about oxygen use and dependence or independence from the need of oxygen. So if you see the term obligate, think about the term obligate for a second. If you're obligated to do something, right, you have an obligation. What does that actually mean? It means that you have to do it, right? I'm obligated to take out the trash on a daily basis, so I have to do that, okay? On the other hand, the term facultative is a little bit different. Facultative means it prefers one way of doing something, but can do that same thing a different way that's less common. So for instance, if you say something is a facultative anaerobe, it's going to talk about how it prefers a certain amount of oxygen versus a lack of oxygen. And I'll get into this in more detail in a second. So, obligate aerobe. Obligate aerobe means it has to have oxygen, right? So, absolute dependence. What does that mean in terms of enzymes, right? SOD and catalase, right? What does that mean in terms of those enzymes? If you have an obligate aerobe, it tells us that obligate aerobes have both SOD and catalase. It's both. Obligate anaerobe means that it has to live in an environment without oxygen. So aerobe means with oxygen. If you put the letters AN in front of something, it usually means not. So anaerobe means without oxygen. Without oxygen. So obligate anaerobe means lives in an environment without oxygen, which makes sense if you look at this test tube right here. The bacteria is growing at the very bottom because oxygen can diffuse part of the way into the media, but it can't get all the way to the bottom. What does that tell you about the enzymes? SOD and catalase. Do you think they have it? Or are they lacking? In this case, they are lacking both SOD and catalase, meaning oxygen is dangerous to them. It will kill them. Excuse me. So an obligate anaerobe, they don't have either of those enzymes, therefore they are going to die in the presence of oxygen. There are a lot of anaerobic microbes out there. Not a ton of them are pathogens. Some, though, some can be obligate anaerobes, but not a ton. Microaerophiles. Basically, this is just saying they use metabolism a little bit, or they use oxygen in metabolism slightly. This could mean they have either low amounts of both enzymes or small amounts of only one of the enzymes, SOD or catalase. Okay. Aerotolerant anaerobe. Basically, anaerobe means without oxygen, so basically these organisms don't use oxygen in metabolism, but they can still break it down into its usable form, which is why they don't die in the presence of oxygen. So if you were to compare obligate anaerobe versus aerotolerant, obligate, notice they grow on the bottom, they do not have either enzyme, which means in the presence of oxygen they'll die. Aerotolerant anaerobe means Anaerobic means they don't use oxygen, but they're aerotolerant, meaning they're tolerant of oxygen. So that tells us they don't use it in metabolism, but they do have the ability to break it down using those enzymes. And then facultative anaerobes, notice the growth at the top here. They prefer using oxygen in metabolism, but if no oxygen is present, they have the ability to survive without it. And they can have typically one of the two enzymes, but it depends upon the organism. So yes, they can manage oxygen in terms of the reactiveness. So they prefer with oxygen, which means they probably have both catalase and SOD, but they can survive in environments with little to no oxygen. Oxygen requirements are very, very important. I promise you, you will see this in terms of these concepts at some point. So here are just some definitions. This is pretty much what I stated verbally, talking about obligate aerobes, microaerophiles, facultative anaerobes, anaerobes in general, aerotolerant, and then obligate anaerobes. So again, this is just what I was talking about when we were discussing that image. Here's the information written down on the PowerPoint. Okay, so I'll let you review that on your own time. I'm going to move on. So if we look at this image right here, it shows us that in the lungs and in the stomach and in the blood, there's going to be obligate aerobes, there's going to be some microaerophiles. For instance, one really good example of an obligate aerobe is in the lungs, Bordetella pertussis, which causes whooping cough in infants. It's an obligate aerobe, so it must be in an environment that has lots of oxygen. Tuberculosis is a facultative anaerobe, so you would think that something like tuberculosis that colonizes the lungs would be an obligate aerobe, but actually it's facultative because the mechanism behind how it causes disease makes it that way. And we'll talk about that later in the semester. But as you get down further into the body, less and less and less oxygen are present, until you get down to the intestines where there's pretty much little to no oxygen. Skin, a lot of people are shocked to see that a lot of organisms are facultative anaerobes, meaning they grow all right, perfectly fine with oxygen, but they can survive without it. Think of a cut, right? The bacteria on your skin, they're aerobes, right? They use oxygen. But if they were to move into the skin, into like a deep puncture wound, for instance, chances are there's very little oxygen in a deep tissue wound. So they have to have the ability to survive without the presence of oxygen as well, which is why they're facultative anaerobes, many of them. And a great example is Staph aureus. So I mentioned that growth requirements include nutrients, they include oxygen requirements, pH, salt concentrations. There's lots of things required in order for microbes to grow efficiently and effectively. Some bacteria can't build all of their precursors, meaning amino acids, vitamins, things like that. Some bacteria have to obtain them as opposed to building them, so they have to import them from the environment. Substances that can't be made on their own are called growth factors. Substances that can't be made on their own are called growth factors. So for us, humans, kind of something comparable to this is there are certain vitamins we can't make that we have to take in order to meet our body's need for these vitamins or minerals. Those would be considered growth factors, right? Now, that's kind of a loose analogy, but some microbes can't build certain amino acids, so they need to get them from the environment. They can't build certain vitamins, so they have to get them from the environment. And all these things are considered growth factors. And the more there are in that environment, the more the microbe can use. An organism that requires lots of growth factors, lots of different growth factors, is called fastidious. Make sure you write this term down. The term fastidious. Basically, the organism needs multiple growth factors in order to grow. Good example would be a microbe that requires a certain pH and a certain salt concentration and a certain oxygen concentration. Right? It can't make pH, right? It can't make the environment. It can't really modify the environment unless there are some bacteria that can actually do that. But for the most part, it can't really modify the pH in the environment. Can't add or take away oxygen, right? That's there or not, or it's in varying concentrations. So these growth factors, an organism needs them to grow. They're considered fastidious. We'll talk about an organism called Haemophilus influenza B, or what we call HIB. HIB, B as in boy. It does not cause the flu. I know the name is misleading, Haemophilus influenza type B. It actually causes meningitis. But Haemophilus influenza is an extremely fastidious organism. It requires something called hemin and NAD+, which is an electron carrier, in order to grow. So where do we find those in our body? We find them in red blood cells. So they grow on plates in media that have red blood cells that have been broken apart, because the red blood cells, when they break apart, release hemin and NAD, and then they'll grow. But if you only have one and not the other, it won't grow. If you have neither of them, it won't grow. This organism, Haemophilus influenza B, has to have both growth factors in order to grow, and go through binary fission and go through their phases, their life phases. Media. So I've used the term media a bunch. Media is just referring to the type of material you use to grow something in a lab. There are two major types: complex versus chemically defined. Complex is basically, you know almost everything that's in it, but you can't account for everything. Chemically defined is like a recipe in a cookbook. It has exactly how much you need of everything. And we're talking down to like microliters, which is it's like half a drop of water. Okay? Think of like a droplet of water and cut that in half, or sometimes even cut that into thirds, and now we're talking about microliters. For instance, in graduate school, the organism I worked with, Lactobacillus pepae, required, when I went to grow it in the media, it required 10 microliters of a special vitamin, a very special vitamin that was really specific. If I made the media and forgot 10 microliters, which is not much, of that vitamin, nothing would grow because it was essential to grow in that environment for that microbe. Now, complex media tends to be media that we use when we don't really know what organisms are going to be present. It's more of like a generalized media. So, for instance, Gloria Bertani, or we call LB media. LB is super general, and it's what I use in the lab to grow pure cultures of E. coli, because it has a little bit of everything. It has some tryptone, which is basically milk protein. It has yeast cells. Yeast cells have stuff inside of them that can be used by the bacteria. It has some sodium chloride. There's some other stuff in there as well. So we don't know exact levels of everything. So we refer to that more as a complex medium. And honestly, when you get your kits, when you get your lab kit, you'll have a little, what we call a dram, or a little vial of E. coli. It'll be a liquid. It'll be a media. It's LB media, this stuff right here. And in it is E. coli. So you're actually getting a sample of E. coli in LB media. Now I used LB because it's pretty general. It's super generalized, actually, to the point where most things will grow. And I used pure cultures of E. coli to inoculate that LB media. And now it's in there growing and replicating and using all the nutrients. And then you'll have access to it within your kit to use it for two labs. You use it for 2 different labs. Whereas chemically defined, some of that is so tedious. But some bacteria, if you don't have it exactly right, they won't grow at all. At all. So it is important we understand the difference. Now we can talk about differential versus selective. Differential media is media that's built to visually distinguish one microbe from another. Differential media is media that's made to visually distinguish one microbe from another. Meaning, you could look at a plate, and if there's different species of microbes on there, they look different on the plate. They are visually different. Here's a really good example of that, blood agar. You will be getting several plates of blood agar in your lab kit because you'll be using them for several labs. And when you plate on blood agar, when you put your sample on there and you let it grow, you can get alpha, beta, or gamma, what we call hemolysis or hemolysis. Hemolysis is the destruction of red blood cells. So you can have alpha hemolysis, beta hemolysis, or gamma hemolysis. Depending upon which one you have, it looks different on the plate. If you have alpha, like this right here, it looks greenish in color. It looks green because the organism only breaks the red blood cells down partially. It doesn't completely break them down. If you have beta, notice it's almost yellowish, just transparent. If you were to hold this plate up to the light in this middle beta hemolysis, you'd be able to see right through the plate because it kills the red blood cells completely. It destroys them till there's nothing left. And then gamma, end here, gamma means growth but no hemolysis. So when I say something as gamma hemolysis, I'm telling you it grew but it didn't break down any red blood cells. If I'm telling you something is beta hemolytic, then it broke down the red blood cells completely. If I'm telling you if it's alpha, then it broke the red blood cells down partially, and it looks green in color. Okay? So, this is a way to visually distinguish different types of organisms. You can tell 3 different... Now, we can't tell species from this, but we can tell that there is a difference. For instance, Staphylococcus aureus is beta hemolytic. Other staphylococci are gamma, and some are alpha. So we don't know which ones they are exactly just from looking at differential media, but we know that if, let's say, we're looking for Staph aureus, that if we had a plate like this, we could say, well, I know Staph aureus is beta hemolytic, so maybe in the center here, where it's beta hemolytic, this is possibly Staph aureus. And then you'd have to continue by taking a sample of that and then trying to get pure cultures in order to identify what the organism is. So this is differential, visually distinguishing. Selective, on the other hand, is basically exactly what it sounds like. It selects for certain organisms to grow while suppressing the growth of other organisms. Okay. So it will support and help provide necessary growth factors to certain microorganisms, bacteria, and then it will also suppress others. A good example of this is mannitol salt agar, MSA, which is another type of plate you'll get in your lab kit. MSA, mannitol salt sugar, it has mannitol, which is a sugar, and then it has roughly 7% salt concentration, so that's actually quite high. Most microbes do not do well in 7% salt. That's too high of a concentration. Unless it's Staphylococcus aureus. Staphylococcus aureus can do perfectly fine in 7% salt concentration. So the point is here that the salt can inhibit organisms that can't handle 7% salt. So anything that can't handle 7% salt won't grow. Things that can handle 7% salt concentration will grow. So it's selecting for some to grow and suppressing the growth of others. This is considered a selective medium. And this is what it looks like. So this tells me, okay, that if there's growth, the organism is selected for whatever, it doesn't matter what side you're looking at. On MSA, it's 7% salt. The growth here tells me, and whatever these organisms are, they can handle 7% salt concentration. MSA also just happens to be differential as well. The sugar mannitol in MSA, if it gets fermented, it drops the pH and it turns yellow, like this. If it doesn't ferment mannitol, the pH stays the same and it stays pink. So you can look at some plates as both selective and differential. And MSA is a really good example of that. MSA is both selective in that it has a high salt concentration, so it inhibits most microbes and only allows the ones that can survive 7% or higher to grow. But it's also differential. Excuse me. It's also differential because we can visually distinguish which one ferments mannitol, the sugar, and which one does not ferment mannitol. Because notice it's a visually distinct. I can visually distinguish between this one fermenting mannitol and this one not. all that's happening here is a simple pH drop. When the mannitol is fermented, the pH drops, and there's a chemical in the media that reacts with the pH, and it'll turn it yellow like this. So blood agar is differential. MSA is actually both selective and differential. And you will actually see that when you use your plate in your lab kit, your MSA plate. One other thing to take note is another example of a selective plate is Sabarod dextrose agar, SAB, Sabarod dextrose. Sabarod dextrose media is used to grow fungi. Bacteria typically do not grow on Sabarod because of a high pH. So, differential, blood agar. Selective, you could say Sabarod selects for fungi to grow and not bacteria. And then MSA, you can say, is both selective and differential. Anaerobic media. So, anaerobes, organisms that survive without oxygen, make up a big percentage of a population in wound infections. So when collecting a sample, you have to be really careful because oxygen kills the microbes before they can even be cultured. So there's media that specializes in environments. Let's say it's a test tube, for instance. There's specific media in the bottom of it that has gotten rid of, or will be able to get rid of, any oxygen that's present. And it'll make it an anaerobic environment. A good example of this is, I'll give you a personal example. When I was younger, 15 or 16, I stepped on a nail and it punctured my foot. And it punctured probably about 2 inches into my foot. Now, there's a lot of microorganisms that live in soil and on rusty nails, most notably Clostridium tetani, which causes tetanus. So when you get cut by something that's rusty or old, or like a piece of wood that's been outside, you usually have to get a tetanus shot because those bacteria get deposited into deep tissue, and then they can grow because there's no oxygen, or if there is, it's very, 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 very little. So when they try to culture it, they have to quickly get the sample and put it in the anaerobic media before the oxygen kills those microbes. And if you've had blood drawn, if you've been sick and you've ever had blood drawn, and they use like 5, 6 different vials, test tubes, some of them you may see are bigger than others. The smaller ones that are a couple inches in height and about as round as a dime, maybe a little bit bigger than that. Those are normal. That's mostly normal media, not too specific. But the bigger bottles tend to be more specialized, and those are typically your anaerobic media. So if you've ever seen them put samples into the big test tubes or the big bottles, that's usually anaerobic media. This is a cool tool that's used in research labs as well as hospitals, called an anaerobic chamber. Anaerobic chambers are neat because they provide a completely anaerobic environment for you to work in, and that allows you to handle microbes that normally you wouldn't be able to work with in a lab. So having an anaerobic chamber is really neat. You put the samples in this little door here, it opens. There's a chamber, You close the door. There's an actual door on the inside of this as well. So there's a little chamber in here. You close both doors. You put the sample in, and then it removes all the oxygen and flushes it with CO2. So it removes the oxygen and then flushes the chamber with CO2, so there's no more oxygen left over. And then you can open the inner door and bring your sample in and work with it. This is what I had to do for my research in graduate school, because the organism I worked with was a anaerobic bacteria. So I did most of my work inside a anaerobic chamber. I put my samples in there and I would work with them in the chamber. Now, it looks pretty cool in an image like this. It looks fun and looks interesting, but I can tell you honestly, they are not as fun after a while. These sleeves, they're all bunched up right now. They're very thick vinyl, and these gloves are very, very thick rubber gloves, almost as thick as some oven mitts, for instance. Or think of gloves that you wear when you wash dishes. Okay, they're pretty thick. So if you work long periods of time in here, your arms tend to get really, really, really sweaty. When you spend 3 hours working in a lab in one of these settings, it's super hot and everything gets all sticky and just it's not as fun as it looks. I promise. It's cool for about the first hour you do it, and then you're like, okay, this is not as fun as I thought it was. However, it is a vital piece of equipment. Aseptic techniques, basically, what is aseptic techniques? Well, aseptic technique, from what you learned in lab, and hopefully you paid attention to this, is basically ways to prevent contamination, right? It's really important to follow aseptic technique because in a clinical setting, you get a sample from somebody. Let's say they have a wound in their arm. It's a very deep wound that needs to be closed. It needs to be sutured, stitches, staples, whatever. You get your sample, and you don't want to have to go back and say, all right, well, my bad, I didn't get a good enough sample. Let's rip open these sutras or staples and get another one. So you really have to be careful, and aseptic technique helps prevent contamination. Wearing gloves, wearing a face mask, wearing a lab coat, not breathing on somebody is really simple ways of washing your hands. Simple ways of following aseptic technique. A really good example is a lot of people want to go into healthcare. As a healthcare professional, you will have to walk into a room. You should be, if somebody, let's say you're working on the step-down unit at a hospital from somebody who just had open heart surgery. If you've never seen it, they don't always close the chest cavity completely after open heart surgery. Sometimes they leave a tube in there for drainage, and it's not completely closed. And just in case they have to get back in there if there's an emergency. Anyway, point is if you walk into that patient's room and you don't have gloves on and you don't have a mask on and you just simply are reaching over them, you're going to potentially contaminate that open wound with some of your microbes, some of your normal microbiota. And it's normal to you, but if it's now on a sick person, someone who's already immunocompromised, then it can wreak havoc over that person. So somebody in that case, you'd have to put on gloves. You'd have to wash your hands, put on gloves, wear a mask, wear probably a disposable, almost like a disposable gown over your scrubs in order to prevent or limit as much contamination as possible because that person is high risk. So it protects people, aseptic technique, and promotes sample integrity. And that's really important. If you get a sample and you don't follow aseptic technique and now it's contaminated, how do you know what to treat the patient with? You get a sample from their throat and you don't put the sample in the media right away. You kind of set it down on the side for a second, on the side of a table, and then you put it once you open the media. You forgot to open it. Unfortunately, that's not good aseptic technique. That sample integrity is not very high because there's microbes just everywhere, and allowing the introduction of those microbes definitely decreases sample integrity, which then in turn, if you go to the lab and they say, whoa, hold on, there's this species and this one and this one, there's like 8 different species of bacteria, then, well, how do you know which one to treat? Right? You can't treat them all, and you don't even know if all of them are causing the infection. Maybe it's just one of them. But because you didn't follow aseptic technique, then you now have a poor sample integrity, and you have to go get another sample. Hopefully, you can go get another sample, and in some cases you can. Some cases you can't. Yeah, samples have to be collected and transported aseptically. We don't want contamination. Using sterile materials is essential. That's why when you go to the doctor, they open up new needles. They everything is almost everything. I don't say everything. Almost everything is single use. Surgical tools are not, but they get sterilized. We'll talk about that later. But the point is most things are going to be single use, and that is because they're sterile. They get used. They're not sterile anymore, so you toss them. Containers that you use to collect samples, for instance, a blood sample, those tubes, those test tubes, are sterile inside. They are sterile. And honestly, one of the best ways to prevent transmission is by using soap and water. Washing your hands thoroughly before and after handling a sample or going in to handle a wound or anything like that. That's the best way to go about it. So isolating microbes in a clinical sample, sometimes you have to isolate it first to make sure that the pathogen is what you think it is. So somebody comes in with strep throat, they swab your throat, they streak it on a plate. Nowadays they have rapid tests where there's just chemical reactions. But before then they would streak it on a plate, they would get isolated colonies which were pure, meaning they were not mixed with anything else, no other pathogen, and they'd pull that sample and they say, all right, yep, it's strep, this is what it is, and then they treat it. Nowadays, it's a lot easier because there's really rapid chemical tests for strep. But still, same concept. For some pathogens, there are no rapid tests. You have to isolate it in a clinical setting. And the streak plate method, that is the most common way to do that, is to use the media, streak heavily in one quadrant, and then sterilize your tool, which is an inoculating loop. Then you move from the first quadrant, you pull some of the sample into the second quadrant, then you sterilize your loop, and then you come back in and you pull some into the third quadrant from quadrant 2. Basically, it's a dilution, right? You're taking less and less microbes as you go around the sample until you get individual colonies growing on a plate. Now, TA showed you last in lab 4 how to streak for isolation using the streak plate technique. This is a perfect example. This is what an ideal sample looks like after you streak for isolation. Heavy inoculum in quadrant 1, pulling some of it down into quadrant 2. Notice there's a significant dilution from this to this, and then going from quadrant 2 into quadrant 3 is even a more significant dilution. And you can see the arrows here in quadrant 1, quadrant 2, and quadrant 3. You're diluting from one quadrant to the next until you get isolated pure colonies. These colonies, all these individual colonies, were derived from a single bacterium, from one bacteria. And now it's billions because you can visually see the colonies. You will be doing this in your lab kit. You will be doing this in your lab kit, which is why your TAs created those videos, because you're going to need to try and isolate some cultures on your plate using the streak plate method. All right. Controlling growth. How do we control growth? Well, there's a couple things we can do. First, we can decontaminate something, which basically means remove or reduce microbial populations for safe handling. So if something is decontaminated, you've removed enough of the microbial population for it to be okay to handle. But it doesn't mean it's sterile. Sterile is very different. Sterilization means there is no microorganism present. If you sterilize something, it eliminates all bacteria, all viruses, all endospores, which I'll talk about endospores. I've mentioned them before, but I'll talk about them more in a bit. And then disinfection just simply reduces the microbial numbers. So if you were talking about spraying down your kitchen counter with Lysol, that is a disinfection method. That is disinfection. You're not getting rid of everything. There's no way to kill endospores with Lysol. So that is a disinfection method. Sterilization is something that you would do with like an autoclave, which I'll show you in a minute what an autoclave is. I have an image in this PowerPoint. So there is a distinction between the two, and it's really important that you understand that. A lot of people throw those terms around without really understanding. So physical ways to control growth, temperature, radiation, filtration. These are 3 common ways to control growth. Cold versus hot, they have different ways of controlling growth. If you put a sample in a cold environment, the microbes will slow down in terms of their growth, which is why we use a fridge for our food. We put it in the food to slow down the microbes from growing and prevent spoilage. If we want to, in a lab, let's say, actually a perfect example is the microbe I worked with in graduate school. Before I left, right as I was graduating, I took my pure culture of my sample. I put it in a specialized media that could survive a really cold environment, and I put probably about 10 vials of this bacteria, this very specialized bacteria, in what we call a -80. It's a -80 degrees Celsius freezer. It's so cold you have to use really thick gloves to remove samples. So at a -80 freezer, we're freezing everything. We're preventing that microbe from growing. It's in the media, it's there, but it's not growing because the temperature is just too cold. And what we can do is, if, let's say I go back, and this is almost 10 years later, 10, 12 years later, I could go into the -80 freezer right now. I could pull out my samples. I could let them warm up at room temperature for a while. Usually it takes about 12 hours to 24 hours, depending upon certain factors. And then I could take a sample from there and put it in fresh, complex media, and my microbe would start growing again. So the point is here is we can use cold to slow down the process of growth. Now, when you are in lab, let's say you're in lab in person, obviously we're not this semester, but lab in person, after you inoculate the plates, your TAs put them in an incubator to have them grow fast for 24 hours, and then they move them to a fridge, which slows the growth back down. Because in a regular semester, when we meet with lab face-to-face, right, we only meet once a week. We only meet on a certain day. So they have to speed up growth by increasing the temperature, which we'll talk about in a second, to get some growth. And then they have to slow it down because, well, you're not coming back for a whole week. So they don't want your plates to overgrow, so they put them in the fridge to slow them back down. Really important. So you can use heat to speed up growth, cold to slow it down. Autoclaving. So this is a machine that is an amazing machine in how it functions. It can sterilize whatever you put in it. And remember, we just talked about sterilization and how meaning sterilized mean there's no microbes whatsoever. No endospores, no capsules, no viruses, no nothing. It essentially kills everything. The way it works is you put your sample into the machine, and these machines, the one in this image right here is a tabletop one. This is, notice these are all dental equipment. So dentist's office will use this. They'll sterilize their tools. Surgical tools that are not disposable will also go into an autoclave and be sterilized that way. So tools like that in a medical field actually get sterilized with autoclaving because it's really the most effective way to sterilize something. But the plates that you use in class or in lab that you'll get when you get your kit. I believe everybody is getting 12 plates. But those plates, the media that is in those plates is sterile because your TAs put them in the autoclave, let the autoclave run, and it sterilized. It killed everything, and then they pour the plates into sterile dishes, and then they let it solidify, and there you go. You have a sterile petri dish full of a certain type of media. Autoclaves work by high heat, high pressure, but it's very important to have what we call not dry heat, what we call steam simply. We call it wet, but the way we describe it in biology is a little weird. But the point is dry heat is something where you don't have steam, and then an autoclave is something where you have steam in addition to high temperature and a high amount of pressure. So 15 pounds per square inch of pressure and steam heat at 121 degrees Celsius. For media, we let it run for an hour in that situation, under 15 pounds per square inch and 121 degrees Celsius. By the time we pull that out after an hour, everything is sterilized. nothing in there is alive, which is what we want. In our department in biology, we have two autoclaves which are really large. They are probably the size of, I'd say 6 feet tall and about 3 feet wide and at least 6 feet long. We have two of them in the department. And those are considered relatively small. In graduate school, where I went to school, we had two autoclaves that were 8 feet in width, 6 feet in height, and 12 feet in depth. So they were huge. They took up the two of them took up an entire room, and they could autoclave hundreds of bottles at a time, which was great because when you try to get your sample, when you need to get samples sterilized, you put them in the autoclave and you just hit a button, and it does all of this on its own. You don't have to sit there and watch it, and it's so effective that it will sterilize whatever your sample is. Sterilization is removal of all things. Now, one of the biggest problems is getting rid of endospores. Endospores, I've talked about in the past, are basically these little areas in cells, and there's only two major species that are genera that actually form endospores, Clostridium and Bacillus. They form endospores. So when conditions are unfavorable, they basically move all the genetic material and all the important stuff to one area, and then they surround it with coat after coat after coat of protective layer. And then the rest of the cell dies, and then those little spores are left over. So it occurs, what are they? They are these spores that have the genetic material of a cell, either Clostridium or Bacillus. Then they occur when conditions are unfavorable. So instead of the cell dying, it forms an endospore. Endospores can survive pretty much anything except for autoclaving. Autoclaving is one of, there are some chemical methods as well, but autoclaving is one surefire way to get rid of all endospores. Endospores are what is what's really concerning with some diseases. So to eliminate them, you have to make sure everything is sterile. For instance, I gave the example of Clostridium tetani, tetanus, when I stepped on that rusty nail. Well, the reason I give that example is because Clostridium form endospores. And if that nail had endospores in it, then endospores would be in deep tissue from the puncture of the nail. And then all of a sudden, they're in a favorable environment, they're going to germinate. They're going to become a full functioning cell again. And then they're going to cause illness. So endospores are very, very dangerous. When we talk about Clostridium tetani, Clostridium perfringens, Clostridium botulinum, we'll talk about all of those organisms and how endospores make them extremely dangerous. Especially since there are very few ways to get rid of endospores. Boiling. So a lot of people think boiling can sterilize water. Again, a lot of people misunderstand disinfection versus sterilization. Boiling does not sterilize water. Boiling will eliminate most pathogens, whether they're bacteria, viruses, or fungi. Bacteria, viruses, or protozoa. Most of them will die by boiling water. Endospores will not. Endospores will survive boiling. Basically, boiling is a way to reduce the risk of contracting a pathogen. It doesn't get rid of them all. And that kind of moves into this next slide where it's pasteurization. You probably have heard the term pasteurization before. With reference to milk or cheese, dairy products. Basically, pasteurization is a really fast way to destroy microbes but maintain the integrity of the product. So when milk undergoes pasteurization, what it does is the company, whatever company it is, if they're working with, depending upon the size, there's multiple ways to pasteurize something. You can use what we call low and slow, low heat for a long period of time, or you can do high heat fast. And when I say high heat fast, I'm talking about 120 something Celsius for 5 seconds or 10 seconds. But you could use, there are many different versions of pasteurization. But the point is to get rid of the pathogens and reduce the number of microbes in that product. Not to get rid of them all. It gets rid of most of them, but not all of them. And the ones that are left over are typically harmless. So pasteurization is a process to get rid of the pathogens. There's a fine balance, and they have to find that balance, because if you heat the product for too long, it will destroy the product. Milk proteins can be denatured, meaning if the temperature gets too hot for too long, the proteins of the milk denature and the milk is bad. It doesn't taste like milk. It's not milk anymore. The proteins are just broken down. So it's important that they know exactly what they're doing. They don't want to destroy the product, but at the same time they want to make sure that they eliminate as many pathogens and microbes as possible in their product. So pasteurization, very interesting process. Dry heat, I mentioned dry heat is less effective than an autoclave, which is steam. Basically, dry heat is ovens, incineration. An example is using a Bunsen burner to heat an inoculating loop. You don't get to do this in lab because we gave you disposable ones for home, because you don't obviously have a Bunsen burner at home. So normally you would use a metal inoculating loop, and you would just heat it every time you wanted to sterilize it in the flame. So it works, but it's not as easy, or it's not as easy as an autoclave. Autoclave, you put a sample in, you press a couple buttons, and it does everything. Dry heat, notice how long it takes. It has to be 170 Celsius, which is 50 something degrees Celsius higher than an autoclave. And in an autoclave, it runs for about 45 minutes. In this case, it has to run for 2 hours at least to achieve sterilization. So dry heat is less effective because of the fact that there's no steam. And you have to make sure the temperature is much higher than an autoclave, and it has to be in that heat for much longer to achieve sterilization. This is why dry heat is really not the best mechanism to sterilize things. Sterilizing an inoculating loop with a Bunsen burner, that's fine, but let's say you have surgical tools, you really don't want to have to use dry heat. You would prefer an autoclave over something like this, like an oven like this. Radiation. So we can use radiation to disinfect or sterilize. There are two types of radiation, ionizing or non-ionizing. And one of them is used to sterilize internal components. The other is for surface components. So if we look at the spectrum of light, at a certain point, at a certain wavelength, you start reaching radiation levels of ionizing or non-ionizing radiation. And depending upon what one you're using, will determine whether or not you're going to use it for something that is inside a container or just on a surface. So ionizing radiation is gamma rays and X-rays. Think about an X-ray for a second. If you've ever had one, or if you've ever even seen one, right? They shoot X-rays through you and they get an image of what your bones look like internally, right? So ionizing radiation can penetrate and destroy internal microbes. Ionizing radiation can be used in food processing. So let's say you get a can of tomato. The company is canning tomatoes, and they get all the cans in a box, and then they shoot ionizing radiation through it, and the radiation will penetrate the box, penetrate the cans, kill any microbes in there, and everything is now sterilized. So this is, right, this second point here, passes through packaging. Things that can't be sterilized through an autoclave can be sterilized using ionizing radiation. And I gave an example of in food, pharmaceutical as well. Medications have to be sterilized. Ionizing radiation can do that. So it's really important that we understand ionizing radiation is powerful enough to pass through packaging and sterilize internal components, internal food products, liquids for medications, things like that. Non-ionizing radiation is typically UV light, and it messes the DNA structure, and we use that for surfaces. So there are these things we call biohoods. Basically, they're cabinets that have a glass door in front of them that slide up and down. And if you're working with microbes, microbes that could be dangerous, you work with them in this hood. Your arms go in, and your face is outside, and there's a glass window blocking, any potential contamination to your face. So you're working in there, and you get done. You're like, all right, I'm done. You pull your samples out. Next thing you do is you flip on a switch that turns on UV light. And the UV radiation, which is non-ionizing, will kill anything on the surface because it damages the DNA. It basically, what thymine dimers are thymer dimers. Basically, if you have thymines in a row in the DNA sequence, it causes them to cross-link, and it forms a bubble, and then there could be no replication, there could be no transcription at all, because these thymer dimers cross-link and create a weird shaped bubble that doesn't allow replication or transcription to occur. Both are effective, but it just depends upon what you're using them for. Here's the biosafety cabinet that I was just mentioning. This front glass piece opens up, slides up a little bit, and then you can work in there. And then when you're done, you flip on the UV light, and it kills everything on the surface. You can use non-ionizing radiation to kill microbes in drinking water. Disinfect surfaces in pretty much anywhere. Operating rooms. This is a biosafety cabinet here. It's effective when you're trying to get rid of things on surfaces. That's the key here. Surfaces. Filters, HEPA filters specifically. So when it comes to things like HEPA filters, they're really useful in getting rid of almost everything that's within the air. Basically, uh, HEPA filters are really designed to get rid of the smallest particulates. And you can see it gets rid of 99.97% of airborne substances. Pores, the holes, are less than 0.3 micrometers. Okay? Micrometers. It does not sterilize the air because it doesn't get rid of everything, but it is pretty close. It is pretty close. HEPA stands for, simply it just stands for high efficiency particulate air. So high efficiency particulate air filters. They just have super small pores, and they have the ability to sterilize, or not sterilize, excuse me, disinfect the air mostly, getting rid of almost all organisms. However, some are too small, and they can still get through HEPA. They can still get through HEPA filters. HEPA filters are found in lots of places in hospitals. So, for instance, some hospitals have what we call negative pressure rooms, meaning the airflow in that room is not like all the others where it brings in air and then it cycles it back out. A negative air pressure room just keeps filtering the air in that room over and over and over and over again through HEPA filters. And then it releases it through a special vent. This is because that patient has something that's really, really easy to transmit through the air, like tuberculosis. So they put them in these negative pressure rooms to prevent air from escaping that room and contaminating any other room in the hospital. HEPA filters are really useful. You can also use filters to sterilize liquids. Pore sizes 0.1 micrometers for bacteria, all the way down to 0.01 micrometers for viruses. So there are situations when liquids need to be sterilized using membrane filters, not autoclaving, not radiation, not dry heat, none of that. A really perfect example of this is antibiotics. If you sterilize antibiotics with autoclaving them, they break apart. The chemical structure can't handle that temperature and pressure. Same thing with radiation. Can't handle the radiation. The chemical structure breaks down. So things like antibiotics or certain liquid medications are filtered to be sterilized as opposed to using autoclaves or dry heat or radiation. A couple terms here. Germicide just refers to controlling microbial growth. They're chemicals that control microbial growth. And then germicides can be microbiocidal or microbiostatic. Cytal means kill, static means inhibit. So if I tell you I'm wiping down a table with a microbiocidal chemical, you should be able to tell me, all right, well, that kills the microbes wherever you're wiping that table. If I tell you I'm wiping down a table using a chemical that is microbiostatic, what I'm doing is I'm getting rid of some of the microbes, but mostly what I'm doing is making it so they cannot continue to grow. I'm preventing them from increasing in numbers. They are inhibited by that chemical. So cytal kills, static inhibits. There are two key classes: disinfectants and antiseptics. Disinfectants you use inanimate objects. Antiseptics you use on living tissue. Okay, I'm not going to really go much further than that with these two classes. Disinfectants, inanimate objects. Antiseptics, living tissue. So that is the end of chapter 7. Hopefully everyone is caught up. I will be posting chapter 7 participation assignment shortly, and then you'll have access to that. And then on over the weekend, I'll post chapter 8. All right, have a good day.